Okay, before you start, let me introduce today's speaker, Arvind Ramanathan. He's a computational biologist in the data science and learning division at Argonne National Laboratory and a senior scientist at the University of Chicago Consortium for Advanced Science and Engineering. And his research interests are at the intersection of data science, high-performance computing, and biomedical sciences. Arvind has published over 30 papers, and his work has been highlighted in the popular media, including NPR and NBC News. More information about Arvind can be found at his personal website. And let's welcome Arvind for today's talk.
All right, thank you so much. So I sort of kept this talk somewhat on a smaller time scale rather than talking for an entire hour. So, if you have questions while I'm talking about it, just, feel free to reach out or put it on the chat window and I'll try and answer it as much as I can during the talk itself. So I want to thank everyone for the opportunity to present. I'm going to talk a little bit about how we are thinking about designing biological systems, and I'll define that a little later. But with agentic reasoning models that we have been building in the lab, right? And none of this work would be possible without the help of so many different people, my collaborators, including Anima from Caltech and Xiaowei and Yunxian, Christian, so many people from NVIDIA that make the hardware and the software interface work, folks at Cerebrus who supported a lot of this large-scale reasoning models and how we can support them. Of course, my colleagues at Argonne and at U Chicago who have been instrumental in doing it. I'll highlight the different people that are playing a role as we come along the talk. The visualization folks that helped a lot with developing what we needed as part of this narrative, the data folks that help us actually build the infrastructure to make sure we can get the data in and out of the global system. Finally, the folks at the robotics side who have been engineering and playing with the system to get us to do what we're doing. Okay, so before I even begin to talk, I sort of want to frame this in the perspective of two very related questions. One is this sort of idea that if we soon have access to, say, an able scientific assistant, right? A person that can sort of help you critique and comment on the ideas that you're building in a way that provides you with very quantitative or measurable feedback, then how would you use this in the context of doing science? The second one is, let's say you have access to these facilities that are now going to be massive industrial scale facilities where you can run experiments that are just designed through your conceptual inputs. So that means that you don't really have to worry about, asking a wet lab person to actually run an experiment, but you can actually describe your experiment. And those experiments are then translated into commands that a robotic center like this would agree and run. And eventually it'll give you access to a lot of data that you can then use to drive, the hypothesis that you're actually thinking about. So if you basically think of these two types of advances that we can make, not that it is here today, there are some places that are doing it, but if you have this sort of advancement in the space, then now the question becomes, how do you evaluate scientific hypothesis, right? And how would you think of your scientific questions itself on a daily basis? Right. So that is the sort of frame that I'm going to use in my talk. And before I actually start that, I'm going to start with the second portion of my talk, which is, trying to understand what is the implication of having these massive industrial scale facilities where you can run experiments through verbal or conceptual development of what that experiment will look like. So if you look at lab automation as such, there is a very rich history for over 70 years or so in terms of how this has been built out. So in 1950s, you saw some analog automation. Computer automation started to make its mark from the 1960s all the way to the 2000s, where you now have very high throughput experimental capabilities that allow you to run potentially like thousands of experiments in a very short amount of time. One of the first approaches towards full autonomy, which is where you use machine learning or artificial intelligence tools to guide an entire experiment, came out in, say, 2009, where the lab of Ross King essentially built this hardware called as the Adam robotic interface that allowed them to question yeast uh, growth under various conditions using a very simple decision tree-like algorithm. Right. So this is one of the first papers that actually showed that you can actually have automated labs that could run experiments in a cohesive manner. And if you see the setup of the system, it's not a benchtop system per se, it's an entire room. And the hardware actually consists of various liquid handlers and various types of other instruments that are put together to measure what you actually consider within yeast as a growth parameter. Right. So if you had these sort of approaches, now the latest advances in AI has sort of revitalized this interest, taking it from laboratory automation towards entire autonomy, which means that the AI is the one that's actually like suggesting what experiments to run, what experiments to design, and how it will run them, and then say whether it can actually run end to end these experiments and be successful in validating a hypothesis. And the hypothesis itself is being generated by the AI in terms of saying, do I understand based on the existing data? Are there some questions that I can ask about the data that will help me, refine this further? So there are several examples of this. One of the first ones that was built was Agentic, or basically an autonomous agent for quantum chemistry that came out from the University of Toronto folks. There's the second one, which is called Robin, which is a multi-agent system for automating scientific discovery. I don't know how many people here have tried out this system called as Cosmos, which is from Edison Scientific or from a place called Future House, where you can actually like have literature search, understanding the sort of hypothesis generation processes actually be done completely in an automated fashion. And then you can come out with like therapeutic candidates using this system called as Robin. And there are various agents that are at play that include Crow, Falcon, and Finch. And they have various different types of workflows to basically do hypothesis generation, lab experiments, and experimental analysis of this. And there are also companies like Lila Sciences or Periodic Labs that are now building what is called as the AI scientific superintelligence, where they are focused on specific scientific workflows that they can sort of automate and actually build through the entire pipeline. Now, given this background at Argonne, we've been like trying to push this from the perspective of like saying, how can we accelerate the discovery process? So what you're seeing here is this lab where there are no humans actually running the experiments per se, but a robotic lab that's actually taking a conceptual description of this very basic molecular biology protocol, something called as PCR or polymerase chain reaction, and trying to implement that within the lab. So on the left-hand side, what you see is a digital replica of the lab itself with all the descriptions of where the ports are and how we should be transporting samples and whatnot. And on the right-hand side, you can actually see the experiment being run. So this work is being read by one of my PhD students, Rory Butler, where in order to get to this part where you can have this automation and AI interplay with each other so that you can actually like run these experiments, starts with this very high level description of a hypothesis that you want to autonomously carry out. And then you use AI and robotics and simulations to sort of close the loop in planning, execution, and analysis of these experiments. So this builds on a lot of the AI approaches in planning from multiple steps, integration across results from various different sources. So in this case, you can see that it's actually integrating information across 4 different instruments that are all very different in their nature of interaction, and then comes out with a protocol and basically decides to how to do it and then executes it. And then you have the automation of experimental protocols that basically describes the robotic steps and the workflows that are involved in it. And eventually you use either self-play-like approaches or reinforcement learning and active learning for selecting the next stage of experiments that you want to do. But in order to be successful in doing this, we needed to sort of democratize this process of actually building these sorts of labs. And that's where we went ahead and built something called as MathSci. And MathSci is basically this library of usable protocols for modular autonomous discovery of science. And this work is led by Noah Paulson with along the robotics engineers that have been building this open source Python-based system that allows you to basically describe any experiment. So any sort of devices, any sort of lab that you can actually run by basically using a set of this very modular ways to describe the experiment itself. It's very simple in the sense that you have good control on your experimental definition with less than 100 lines of code that's written like in human language, like YAML or Python. And then you have a scalable backend that allows us to scale it from just a single device all the way to multiple laboratories. And it's been applied to various domains now. So there are examples from biology, chemistry, and quantum that you can do. And it's sort of being maintained in a vendor agnostic way, which means that we don't really care about the devices that are in your lab. As long as you're able to write the libraries that allow it to interface with the instrument, you should be able to run it anywhere. And what it can do is basically coordinate a number of these autonomous workflows across these devices in a way that you don't have to worry about where the instrument is sitting and how we are running it and so on. So it basically manages the entire collection of data, monitor the experiments via this interactive dashboard, and it also collects and publishes data and logs that basically tell you whether an experiment was successful or it can self-correct on its own in terms of what it wants to do. One of the first experiments that we sort of ran was to design these antimicrobials. And the challenge there is fairly simple in the sense that we have a list of these antimicrobials that we know are in the literature. So there are about many data sets that are available for this. That becomes our training data. Then you use a protein language model to basically understand the similarity across those data sets to say whether you are sampling from that. What we want to do is, given a particular type of bacteria, we would like to suggest mutations on those antimicrobial sequences such that they actually become more functional in the lab. And from the point of view, this is a very baby step in terms of running a full self-driving lab, really speaking. And the only portion where we don't have this power of the self-driving lab is for ordering peptides, because that's something that is financial and not controlled by us physically. But the rest of it is actually like done entirely in an AI model fashion. So you can run this end-to-end workflow as long as you have the peptides available. But the whole point is that we could actually design these antimicrobials and run them in the self-driving manner without any human intervention. So how is this achieved in the lab? So in this context, we actually build what are called as embodied agents that can generate the lab code that is needed for it to run. So in this case, you say, I'm going to do this particular task. It seems like the logical step for running this, you can see the prompt at the top, is to prepare the master mix for the PCR reaction, which involves combining various reagents in specific volumes to create that. And so that is the task. If that were the prompt, the agent is actually inspired by this workflow that was done for gaming. I don't know how many of you have played Minecraft in the past, but Minecraft is a game where you can actually create scenarios on your own and like learn to have new skills and do those types of things. And one of the first examples actually came out from Anima's group where they were building this embodied agents for evolving new scenarios for running embodied agents for running games, computer games. So we use the very same approach. In this case, you have an agent that basically takes into account the task prompt and generates the code by first decomposing the task into various set of tasks, subtasks. So you have the main task, you break it down into these steps, task 1, 2, 3, 4, 5, 6. And for each of that, we are writing candidate code that go into a simulation engine and allow us to test whether each of those code bases are actually working. And that contributes to the level of goal tracking that we can do. So it's maintained in that loop to kind of say, okay, whether I'm doing the right thing in terms of tracking the end goal that I want to do, or if I'm doing something that is wrong, it has to self-correct itself based on the code that is there. And if the code is verified, it goes into this skill set, which we can draw upon later using retrieval augmented generation and so on. But once you have this code skill, it's put into this database, and then we refine the code for that specific protocol that we are running. So in some sense, it can adapt itself to any new protocols that are given to it. It's not that straightforward to basically like learn an entirely new skill, but we can still provide it with the sort of self-execution loop that allows it to basically like understand if it's doing the right thing or not. So here is a video that just shows you how this runs. So this agentic implementation of the workflow is broken down into 3 agents here. Basically, there's a verify agent, the planning agent, and the workflow agent that basically generates the step, looks at the different instruments that are present, and then said, okay, when I'm running it, I attempted to transfer a plate on this Heidex Geraldine system, which is a plate reader, where the lid was actually not open for it to transfer the plate, which is a wrong thing to do. And then it decides that, oh, I made a mistake. Let me correct that mistake. And it'll start self-correcting that process of the workflow, basically coding that on the fly and generating that step in such a way that it can actually like go and self-verify and said, okay, now the plate reader was open. I can transfer the plate and I'm actually successful in completing that protocol, right? So this is work led by Brian Su and Priyanka, who are in my group, who have been like basically experimenting with this. Now we can actually do this with all the frontier models as well and see how successful each of the models are. I'm not going to talk about them here, but the idea is that once you have these sort of, simulation protocols, you can always find ways to refine how you're running the skill sets or how you're like refining the runs on, on the fly, really speaking, so that you can actually get every time a runnable protocol end to end being executed. So that basically tells you how we have been thinking about our self-driving labs. That corresponds to the first portion of like saying we can actually scale this in some manner in a lab. Now let's talk about how we want to use this in a real example. So in this case, one of the things that we wanted to do was to target these proteins that are called as disordered proteins. So why are we interested in them? These are proteins with which represent over 30% of the human proteome and are sort of very important drug targets. But the unfortunate problem is these proteins don't have a stable tertiary structure, right? They have very high levels of flexibility. So for this example, I'm going to use this protein called as P53 that you see here, that's labeled in the various colors. Within P53, there is a very small segment of this protein, which is from residues 356 all the way to the end of the protein, which is about 370 or so. And in this span, it actually interacts with over 30 different proteins. Actually, P53 interacts with over 300 proteins, but this particular span is known to involve itself in over 30 different complexes. But in each of these complexes, it adapts a completely different structure, secondary structure. In one case, it's actually an alpha helix. In a second case, it adapts a beta strand. And in the third and the fourth cases, you can actually see it forms a loop-like structure that's very important for the interaction to be stabilized. And what is interesting about these proteins is that about 65% of these proteins are involved in some form of diseases. And we largely want techniques that can allow us to drug these undruggable parts of the genome. This is not only restricted to human genomes per se, we are looking at other types of proteins as well from bacterial and viral and fungal pathogens, especially in the context of infectious diseases. So these are very important signaling proteins that we want to target, but there are no tools and techniques out there to do this. And that's a place where we decided to say, okay, let's build an agentic reasoning system that allows us to do this in some scale. So the StructBioReasoner is one of the AI co-scientist that allows us to do this end-to-end generative design of biological modules. So in this case, your user, who is you or me or anybody, can basically say, I want to describe a high-level goal for the scientific task. So one thing might be that I have an enzyme of choice, I want to increase its thermostability, which is one side of the story. On the other hand, let's say I want to design a binder that targets the specific protein-protein interaction. If you're a scientist, one of the first things that you normally end up doing is to basically like understand it from the perspective of literature. So you have these agents that allow you to now basically go after this, scientific literature and do this at scale. So we have developed tools inside the group for doing this. One is called AdaParse, which is basically an adaptive parsing strategy that allows us to parse all these PDF files and get high fidelity extraction of text and other modalities as well. And the second one is a hyperrack system, which basically allows us to do scalable retrieval augmented generation. And it's adapted for state-of-the-art open models so that you can actually get answers that are quite useful across the different domains that you're interested in. So it might be chemistry, it might be physics, it might be biology, and we have all those sort of represented in the data. Now, once the user specifies the goal, it goes into the literature exploration mode and then gets you answers. But in order to do that, we sort of decided that we can run it across supercomputers in the country. So this includes Aurora, which is at Argonne, and Polaris, which is also in Argonne. But on NERSC, which is a system called Perlmutter, and Frontier, which is also one of the largest supercomputers on in the world, which is at Oak Ridge National Lab. So we can actually basically instantiate this workflow in a completely automated fashion across these different sites and use something called as Academy. So Academy is something that is developed by Ian Foster's team here, where they have an agentic platform that allows us to basically instantiate these models across different locations and then basically allow them to run across these supercomputers while not worrying about how data transfer is done, how the agents are coordinated and coupling to each other and so on. So that allows us to do something very, very modular and very conceptual, but Academy takes care of running it across the different systems that we care about. So this adaptive parallel parsing of PDFs allows us to assimilate scientific findings. And I'm going to talk about a specific protein called NMNAT2 here. NMNAT2 is a protein that's involved in cancer through the regulation of the levels of amount of ADP in the cell. It's a very key enzyme, but about 75 residues in this protein is completely disordered. And so nobody really understands how to target this protein, because any drug that you design is all going to target the active site, but there are many different proteins that have the same level of function like NMNAT2, including two isoforms called NMNAT1 and 3. So you don't want to accidentally hit the other two proteins. And therefore, the only place where these proteins are different is the disordered regions, and that's what we want to target. Now, using our adaptive parallel processing system, we can actually parse about 78 PDFs per second on this system called Polaris. And it's also achieving about 92% accuracy without compromising the quality on the PDFs that we can process. And this was published in MLSys last year, where we can see that the AdaParse can actually process text, images, tables, and references to cover all the modalities within the scientific data. Once you have the scientific data that you really care about, the representation, we now have a way to retrieve the information that efficiently scales up the entire process of connecting the knowledge. And so on this particular example, we have indexed over 3.6 million full scientific text articles by using question answer sessions that allow us to basically get back answers that we care about. So given these two approaches, we can now use this to basically have a chat agent that basically allows you to understand all the interactions that are involved in the NMNAT2 interactions in the cancer pathways. So just to see how this looks like, let's take a deeper look at it. So the hyperrag agents basically goes and says, okay, I want you to identify all the NMNAT2 interaction druggable interfaces. And then it goes and tells you something about the protein. It tells you all the interacting partners. It tells you the druggable interfaces as well. And it also tells you all the citations which are involved. It also tells you, more importantly, what residues are involved in those interactions. Now, if you get these sort of residue interactions, these become natural, part of translating them into something that basically can allow us to fold these proteins using AlphaFold. So if you were to use AlphaFold naively on this protein and say what you see, you would get something where the disordered region essentially looks like a long loop without any order. It doesn't have any secondary structure. But if you use this sort of information from the different papers, you can now get more details with respect to the 3D structures and how it should be modeled. And we use these as constraints for these protein structure prediction problems, which allows us to model the entire interaction network. And I'll show this in a little bit where we show the video of how the interaction network really looks like. So once we discover these NMNAT2 interactions in cancer, we can now actually understand with respect to P53, there are two primary hydrophobic interfaces that it actually interacts with. So this is all coming from literature that basically says that this particular protein has these interactions and you're using this information automatically to define the structure. So here is the interactome of NMNAT2. In this case, I'm only showing you a subset of the 45 proteins that it is interacting with. We run these simulations in an automated fashion, which means that the agent actually knows that these are the simulations it needs to run. It folds them using a model called as CHI, generates the secondary and tertiary structures, builds them off as simulation environments. And in this case, I'm looking at the NMNAT2P53 complex, where it shows you exactly what are the interactions and the residues that are involved in the binding. So what is more interesting is that that particular hydrophobic interface is very stable, and you can see across the simulations, these are probably about 50 to 100 nanosecond simulations, multiple replicas of it so that we get statistical significance. But it allows us to basically understand something about the interactions and so on. So what is actually pushing the entire system is this BioReasoner framework that's being developed by Brian Su and Nick Chia. And this system actually uses a question answer framework that's generated using a framework called as Argonium. And that's actually about 150,000 highly curated data set where we can filter it for high uncertainty questions. And from those uncertainty, this ones, we can actually have a self-training loop or self-play loop using reinforcement learning so that we can actually get the correct rollouts for how the reasoning chains should look like for training that particular model. And that supervised fine-tuning training is now, data is now collected, and it becomes part of our BioReasoner framework. What we have seen with this is that even smaller models like DeepSeek 8 billion parameters can approach the function of OSS 120B. In this case, the yellow line represents the OSS 120B, which is one of the largest open source reasoning models from OpenAI. And we can see that we are closely approaching what OSS 120B can do when it is actually run through this entire framework for training. And in this case, we are now asking questions via the journals, which is kind of interesting from the point of view of how the reasoning framework should work, where it has access to a few easy questions. So some that have medium complexity and some that are extremely complex to answer. And this model can actually do that by being trained in a specific manner. Now, let's say I get all my interactions and interactome that actually tells us what these key sites are and how I use that. I can now instantiate the next step of my workflow, or the agent basically decides that it needs to run something called as a molecular binder design agent, which uses protein or peptide design language models to basically suggest what a peptide should look like for binding them. And then it uses the same framework for running MD simulations that you saw previously that allows us to refine the designs using these simulations. So that's what we're going to see now. And so in this case, you saw the NMNAT2 P53 complex from the past, and now it's now diffusing various peptide binders to the active site. And that site is actually shown in the purple color there. But what is interesting is the diffusion actually follows this process where it is going to all the places other than the peptide that it needs to focus on. But after a while, it starts to actually focus on the right region because it knows that this is the region that it wants to target. And now it can actually look for good backbones, filter out the sequences that have bad backbones automatically, and then look at ways in which these interactions are held stable. And in this case, you can actually see that there are some hydrophobic and hydrophilic interactions that form the basis of this interface that allows us to basically say, okay, I can reason that these interactions are making it more stable. And those interactions are the ones that I'm going to pay attention to in the next few minutes of my talk. So how does this scale across our system? So really speaking, we can design binding to scale to up to about 200 million designs every day, which is a lot of work. But you can see with respect to the different lengths of the amino acid sequences of the protein and so on, we are close to ideal in terms of scaling. And even with 1/10 of the machine on Aurora, we can get to about 800,000 peptides. This includes both the design side, the inference piece of this, the MD simulation side, which are put together in one workflow for the design agent to work. But how does this take into account some of the experimental data? So we have been working with a bunch of our collaborators in Chicago, Brian Dickinson in particular, where they have a technique called the SPANCs, which basically allows us to take very large phage variants and then design these binders for design and using these to basically like look whether they bind or not. So in this case, what you see here is that there are various scaffolds that they have used to basically identify, say, about 10 binders or so that they have. And we use this information to actually feed back into the reasoning agent to kind of fine tune itself in terms of generating peptides that might have higher affinity to this, right? So I'm not going to talk a lot about that today because that's a lot of experimental data and how it feeds back into our preference optimization strategies and so on. But once that happens, we can now identify potent inhibitors that are now targeting that particular interface by stabilizing it with a bunch of salt bridges and hydrophobic interfaces that allows it to basically stabilize that peptide protein interaction. Now, this basically lets us say that we can now get these into the lab for basically testing. So in the first part of my talk, you saw this little piece where we said that, okay, we could take a protocol, translate it, and run it in the lab and say we can run these experiments. But now we can scale this entirely to an entire factory scale facility where you can actually like potentially theoretically also describe the designs across 200 million peptide design experiments in one scale. And so what Rory has done here is basically to simulate that at a virtual facility scale that basically tells you how he can run these across many, many, many instruments and how many instruments he would need access to in order to run that. So in this case, he has basically like optimized it to be about 100 instruments that are shown here. But that basically allows us to say how many instruments we would need to resource and whether those instruments are available and where we can run them and so on. So that kind of ends up completing the loop from where we started, which is just a hypothesis of saying that I want to target this particular protein, all the way to an experimental loop that connects everything from simulations, actual experimental data that adds this feedback to these models to like kind of play with the reasoner to say, how do I reason about the next step of my experiment and so on, and eventually lead it to the next stage of how we want to test that. So these projects are under the auspices of what is called as the US Department of Energy projects, especially what is called as the Genesis mission. I don't know how many of you might have heard of it, but the Genesis mission is a large-scale DOE investment in terms of AI and particularly in biotech. There are several projects that are going on in the lab. If you're interested in talking about any one of them, you're always welcome to do that. So the project that I showed you about this co-scientist workflow, this is something called as the IDEA, which is an intelligent design assistant that we are building. There's also research that's going on in terms of understanding extremophiles and how they process metals in various environments as part of a project called as MEL-3. And the orchestrated platform for basically like running these experiments at scale across 3 different facilities in the country is being done under the Opal DOE project. So basically what I showed here hopefully is like a summary of how we are using AI-guided design reasoning models that can be quite powerful for biosciences. I showed you an example where we are doing biologics design, but the same thing holds for enzyme discovery, design, and optimization, where we are integrating a large number of these tools, both bioinformatics, non-bioinformatics related tools, molecular simulations, surrogate models, and others that are going into these platforms for doing this sort of research. And it's also laying the foundation for what we think of these autonomous labs of the future can do from the perspective of digital twins that can actually run protocols and have them run successfully in the real world. And then you can actually evolve complex protocols that are self-improving strategies. But what we have also seen is that they require better world models for these AI models to be more powerful. And there are some limitations in terms of where the reasoning models need better mathematical foundations because they're just doing a very brute force approach of seeing whether something works or not. And then there is also this aspect of scalability and hardware, software co-design that needs to happen with respect to robotics, which is also something that we are in the process of talking to vendors and trying to figure out how we can actually run them at some scale. So with that, I'd like to acknowledge a lot of the folks that made it work. This is including the funding agencies from DOE, ARPA-H, and the Coalition for Epidemic Preparedness Innovation, CEPI, and the computing time that was actually allocated to us from the Algon Leadership Computing Facility, Cerebras and NVIDIA for donating their computing time to do this work. And all the data models and code are available, so you can actually download and play with them if you're interested. So with that, thank you.
Thank you so much. Any questions?
Hey, Arvind, great talk. I had a question about the mathematical challenges or the better math that you were talking about. Can you talk a little bit about what that means for the protein language models?
I mean, within the context of, I don't think they're particularly related to protein language models per se. I think they're more related to the fact that you have representations that are built by these models that now need to be translated into something that the models can sort of understand from the perspective of, causal reasoning, which means that when I make a mutation to this particular site, how does it affect the particular property that I'm interested in understanding? So for example, let's say I ran my design protocol for an enzyme where I found this enzyme is highly stable at 75 degrees centigrade, but the language model doesn't help me reason as to why that particular mutation led it to be more stable at like 75 degrees centigrade. All I can say is that this is the design that came out, and even if I go back and look at all the attention parameters and so on, it doesn't give me a causal reasoning for saying these were the reasons why this became more stable, or these were the reasons why it became less stable in that particular environment. So there's more work to be done there in terms of how do we understand the math side of it from tensors that tell us whether these, the foundations that we are finding with respect to the attention parameters can help us identify the causal relationship between what the property of interest was and how the design actually was suggested. So probably that requires a lot of thinking through how the attention models are making reasoning choices. Or they might not be doing any reasoning choices, but they might be underlying the correlations. But from correlations, we need to move on to something like causation, which is what I think I was referring to there.
All right. Yep. Thanks.
Hi, thank you for the great talk. I really like the part where you introduced the automated lab. So I'm just on a high level curious, what is the current status of these automated labs? How do scientists work around that? Are there any major breakthroughs about agents controlling those automated labs?
What do you mean by agents having a breakthrough? Meaning, in a particular experiment, do we reduce the amount of cycles that we have to do for the experimental design? Yes, there are some examples of that, especially in the context of antimicrobial design, which is a case where I can talk a little bit more confidently because that's coming out soon. That's a space where originally we had a lot of these proteins that were, or the peptides that we were designing were looking like alpha helices and beta strands really. But curiously enough, one of the peptides that it generated was actually disordered, right? And then we had to do a whole bunch of simulations to kind of understand whether those disordered things were actually working in the lab and why they were working. And that was a case where it was kind of interesting to see that the agent decided that, okay, I'm having fun exploring this new space of these materials. But I don't have anything more than a particular example where that sort of a thing was happening. In terms of experimental cycles and so on, like how much time it saves, I don't think it is currently there to argue whether it is saving time for the scientists in doing certain things. Obviously, the scientists actually enjoy a lot of this, which is all very regular, and they can have access to their data in a more, platform-oriented way automatically and so on. But again, remember, this is not integrated into the LIMS systems and things that they're more used to using in the lab, right? I mean, anything that we do like this is going to also break down anything that they do on a normal basis. But I would argue that that is still not a bad thing, right? People have had um, uh, more curious cases where they've seen that the protocols actually are more robust, they are more reproducible, they actually end up becoming, um, you know, much more valuable to them because they don't have to like re-implement a protocol every time that they're trying to run on the robotic system. And the nice thing about what we are doing with the modular platform, like MatSci platform, is that it lets us do things that are completely, um, you know, uh, transparent sometimes to the type of instruments that might be there. So I might have two different liquid handlers. I might have a Hamilton liquid handler versus a Hudson liquid handler. Both have various different capacities that they can do their experiments, but the code is the same. All I have to do is change the liquid handling system to it to be a different one, and I'll get still the same results that I care about. So again, consistency, reproducibility, and the flexibility to run across multiple platforms. And of course, now we are trying to do this across two different facilities. So, as long as I have the same standards of preparing the samples, I can run a connected experiment between the two facilities without ever having to like see that facility. All right. So
That makes sense. What do you think is the major bottleneck right now for us to like push forward that the agent can automatically make some actual discoveries?
Where do you want me to get started? I mean, every portion of the pipeline has its own set of challenges. So, for example, let's take the publications, right? Not all publications are written in the same format, right? So, if you talk to Carlo or if you talk to Ozan, they can give you horror stories about how it is hard to parse these PDFs to get the right sort of representation, right? So, that's one bottleneck. The second bottleneck is many of the codes that they say will work might not even work in the first place, which means that the agent has to rewrite or redo the code that they're claiming in that particular paper to see if that is actually like plausible or not. Right?
Mhm.
We have seen those cases as well. In some cases, the agents are quite, I would say, capable of writing their own code to make it work, right? And that's another space where we have seen progress, where we have seen that the code written by the agent is probably more modular, more reusable for various types of things. Now, we are not in the process of like archiving the memories of these agents as well, which is something that I'm like very interested in hearing ideas about how to do this. Uh, but they evolve a lot of things inside the workflow, which we had not really thought through before. And they've done some very interesting things as well, right? I don't know if I'll put them in the context of, new knowledge, but it's also like things that they end up doing on their own. But that is a bottleneck because sometimes if you have to do that, you have to basically pause your workflow, get that implementation done, test it out. And if the agent is the one that's doing it, and if you're not interfering with it, you can get, you don't know what you got. And so it's very hard to say whether you have the right answer or not. So that's another bottleneck. Perhaps the more important bottleneck is the fact that we don't have facilities that are really scalable right now, right? So the simulation aspects of it, what you're seeing is basically trying to tell you that there's a way to principally reason about how we should think of scaling these facilities in the lab and what we can do. But we still don't have a virtual or a real facility where we can run these experiments at some scale to understand what implications we have or what bottlenecks we would face. Perhaps the bigger bottlenecks are to say, okay, how can the experiment actually accelerate protein production or purification, which I care about deeply, or cell preparation, cell line preparation, which is again very deeply involved technical work, which possibly we don't have solutions for these very much right now, which all have to take time. But if they do come in place, they'll definitely change the way we do science.
That's really helpful. Thank you.
Great. I have a kind of question on the bioreasoning model. I think the performance end up getting very high. I was curious, like, maybe I'm not familiar with the benchmarks here. Like, can you just explain that a little more?
Could you repeat that question once more?
You had one figure where you fine-tuned, like, I think it's called a bioreasoner, but the accuracy was approaching like 100% essentially.
It was not no, I don't think it's a proven.
Okay. Maybe I'm just
Coming to
misreading it.
100%. No. I think we are far away from 100%. I mean, if you do that, I'll be very happy, but no, I don't think we have approached 100%. I would also say that it's close to 95, 96%, but that's also with the OSS 120B, which is more capable. And it's as
Oh, I see. So I think I misread it.
yeah. The
But I guess like
argument is that.
I think in general, like maybe can you comment on these benchmarks a little bit? Like, do you think this actually translates to more capable agents for other tasks that you are working on?
I believe so. The thing is, we have also not tested some of this with tools right now, right? So this is still based on the way that you normally train the reasoners to basically use text as the primary modality to do it. But if you have code plus tools that can be called on, I feel that we could shrink this even further, right? So a model like DeepSeek 8B would probably be more accurate as well and more sensible once it has more of this access to tools and compute code that it can write. So that's something that we are actively working on, right? There's a lot more progress to be made there. But I think this will definitely translate to other reasoning as well. So, Brian might have more ideas on this, but he's also working on a chemistry data that's similar to this, and he's probably also looking at some physics as well. All right.
Great. Do we have any other questions? Okay, if not, let's all thank Arvind again. Thank you so much. This is a very amazing talk. And for people who have been watching the seminar, this is our final seminar this quarter, and we are going to start again in the winter. And stay tuned. We have a great line of speakers for the winter quarter already. Thanks everyone, and see you next quarter.
Thank you. See you all.